In this video, I want to talk about the ways in which we can visualize neurons. How can we stain the brain to learn about the fine structure? I'm going to start by reminding you that even an unstained brain has some structural differences that we can see. So for example, in this brain, we can see the gray matter and the white matter. And today we know that the gray matter corresponds to cell bodies and dendrites, and the white matter corresponds to bundles of myelinated axons. But what I've just told you here about cell bodies and dendrites and myelinated axons is not evident when you examine unstained tissue, even if you use a microscope. In order to learn about the cellular makeup of the tissue, it was necessary to develop stains. So what I'm going to do for the rest of this video is walk you through some techniques that people have used to visualize neurons, and I'm going to do this more or less in a historical order. A good place to begin is with the work of Franz Nissl. Now, he was a graduate student in a lab where there was a competition to develop the best stain, and the stain that he came up with we now call Nissl stain. Nissl stain uses a basic dye to stain RNA and DNA a purple-blue color. So remember that RNA and DNA are acids, ribonucleic acid and deoxyribonucleic acid. So by throwing on a base, it has a high affinity for this. Now Nissl had no idea that DNA existed or why this stain worked. But what he did know was that when he took it and he stained sections, that it differentiated structures. So for example, in this image here of a macaque brain, we can see that the outer portion of the cerebrum is this purple color, and then there are these white parts. If we zoom in a little bit more, we can start to see even finer structure. So for example, here's an area here where there seem to be some layers of purple staining. This is the lateral geniculate nucleus. Here's a discrete structure where again there's this unusual kind of staining pattern. This turns out to be the hippocampus. And here, all the way around the outside of the cerebrum, we have the cerebral cortex. And if we go and we zoom in on one of these areas in even more detail, you can see that even here in the areas that stain purple, it's not uniform, and that the staining appears to be these discrete dots that are the right size and shape to be cell bodies. Let's zoom in even further, and even further. And now I hope you can see that the purple dots really do correspond to structures that look like cells with nuclei. Here I've outlined one of these purple stain structures. So this stain allowed neurologists to gain a lot of insight into the organization of the brain and see that it wasn't uniform. There are also stains for nerve tracts, and I'll give you an example of one. This was developed by Carl Weigart, who is a contemporary of Nissl's. He actually was in the same competition. He lost to Nissl. But his stain that he developed is called the Weigart stain for myelin. So I think the best way to see how Weigart stain works is to compare it to a Nissl stain section. So these are both brains from macaques, and they've both been stained. And you can see that what is blue-purple in the Nissl stain appears relatively light in the Weigart stain, and what appears white in the Nissl staining is black in the Weigart stain section. If we look in more detail at this, we can see many of the same structures. So again, there's this discrete area here, the LGN, lateral geniculate nucleus. Here's the hippocampus, and here's the cortex. But we also see areas where there's a lot of black stuff. And I'll zoom in on one of these black areas. And now we can see that there are discrete patches where there is not a lot of staining, where it's fairly pale, but it's also surrounded by this black material. And zooming in even further, we can see that this black material appears fibrous. This is the myelin sheath that's wrapping around the neurons. Here's another way of staining nerve tracts. This is silver staining. Now here I've switched to a section of a mouse brain. Silver staining involves taking the tissue and impregnating it with silver, and the silver binds to proteins and it penetrates different organs in the cell at different rates. If we zoom in on this and we look at the structure here, you can see that the brain isn't uniform. Zooming in even further, we can see that there are areas where, again, there appear to be cell-like structures, that what we would now today call cell bodies, but there was still this fibrous network out here that didn't seem to be cellular. And there was a debate in neuroscience about what the nature of that fibrous material was. It was not clear how the things that appeared to be cells and how the fibers were related to one another. Answering this question did not become possible until Camille Golgi Italian anatomist, invented a variation on silver staining, which today we call Golgi staining. Golgi staining stains only 1% of the cells, so if you think back to the silver stain sections, they were purple everywhere. Well, with Golgi staining, they'll mostly be clear with relatively fewer spots, and the cells that stain do so completely. Now, Golgi used this technique, but even more importantly, Santiago Ramón y Cajal, a Spanish neurologist, used this technique and applied it to brain tissue. And what Cajal recognized was that the fibrous extensions were actually part of the nerve cell. And we can see that here in these images where you can see the cell body, but you can see that the fibrous extensions are continuous with them. I'm going to show you some examples of Golgi staining. Now these are sections from mice, and they're Golgi stained, but they're also counterstained with a Nissl type dye. So the blue corresponds to a really concentrated layer of cell bodies. Golgi staining allows us to see different morphological classes of neurons. So for example, in the cerebellum, there's a cell type called the Purkinje cell, where there's this huge, extensive network of dendrites. And this cell here that I've shown you, I took from this image over here, but you can see that it's not the only Purkinje cell. And so this one morphological type of cell is found in the cerebellum, and it's found in a repeated array. It's not the only type of cell in the cerebellum. We also have other smaller cells. What Cajal recognized was that a given region of the brain might have 5 or 6 stereotypical cell morphologies that are found over and over and over again in that region and across species. I'll give you another example. Here is a section of the cerebral cortex from a mouse, and again, this is Golgi stain, but then counterstained with a Nissl stain. We'll zoom in on an area. And what I hope you can see is that the morphology of these neurons is different. We call these pyramidal neurons because of the shape of the cell body being sort of triangular. Cajal and the neuroanatomists that followed him were able to use these techniques in order to learn a lot about the cytoarchitecture of the brain. Given that neurons seem to have these long axons that projected from one area to another in the brain, the next question was, how are the different brain areas connected? Where do the axon tracts actually go? For any particular brain region, where is it getting input? And there are a number of different ways to do this kind of tract tracing. The earliest technique was surgical. You would take a scalpel and you would actually sever the nerve cell, and then as that cell degenerated, it would leave cellular debris behind that could be stained, and we could find that debris in the anterograde direction, so towards the terminals, as well as retrograde degeneration towards the cell body. Another tract tracing technique involves injecting labels. So in this case, labeled amino acids can be injected near the cell body. The cell takes them up, incorporates them into proteins, and then as some of those proteins are transported down the axons to the terminals, we can fill that cell in the anterograde direction. It's also possible to do retrograde labeling. So we can take proteins or viruses, put those near the terminals, and axons will take up those substances and then transport them back to the cell body. Sometimes the substances that are taken back go only within the neuron itself. For example, if we inject cholera toxin out at the terminals, that will be retrogradely transported back to the cell body, but it will only still that same cell body. In contrast, if pseudorabies virus is placed out near the terminal, that will be transported back not only to the first neuron, but even beyond that. So you can have polysynaptic tracing, and that can happen in the retrograde direction as well as in the anterograde direction, depending on which virus or substance is used. With the discovery that DNA is the genetic material in the cell, it became possible to exploit this knowledge in order to birth date neurons. To explain how this works, I have to remind you that neurons are long-lived. Most of the neurons in you were generated between 5 weeks and 8 months of embryonic development. And so human brains may contain neurons that are 20 years old, or 40 years old, or 100 years old, depending on the age of the individual. In addition, mature neurons don't undergo mitosis. Once they've divided and differentiated into a neuron, they will not replicate the DNA again. This means it's possible to birth date neurons by incorporating a stable label into the DNA at the time of the cell's last division, and the labels that are typically used are either tritiated thymidine or bromodioxyuridine. Thymidine is one of the nucleotides that makes up DNA. So when you think about DNA in A, G, T, and C, the T is thymidine. Bromodeoxyuridine is a thymidine analog, and when the cell is replicating the DNA, it will grab bromouridine and put it in where a T should be because it can't tell the difference. So we can take an animal and then inject it with a pulse of labeled nucleotide. And if we're interested in birth dating neurons, and remember they're generated during embryonic development, usually this involves taking a pregnant animal and then giving it a pulse. And then you wait for the animal to have its offspring, you sacrifice the animals, and then you can examine those brains for the location of labeled nuclei. The results look like this. This figure shows us birth dating in the visual cortex using either tritiated thymidine or BRDU. For the tritiated thymidine, you develop this using autoradiography, and the label appears as speckles, silver grains. For BRDU, this is usually visualized with an antibody, so here's a brown stain showing which of the neurons were born whenever that pulse was given. In the 70s, it also became possible to couple physiology and structure by making recordings with dye-filled electrodes. So people have been doing electrophysiological studies and inserting electrodes into neurons in order to look up their activity for years. In this variation on the technique, the electrode's not only filled with a saline solution, but also with the dye that can be injected into the cell. Here's an example from work by Owen and Grace. Their research focused on a group of cells in the nucleus accumbens and looking at how the activity changes in response to amphetamine withdrawal. And after they'd made the recording, then they would fill it with dye so they can visualize it. So again, here's this accumbens neuron visualized with the lucifer yellow. It's also possible to react this dye so you can get a permanent preparation, which is shown here. As molecular and cellular biology advanced, it became possible to use other techniques. A widely used technique is using antibodies to label specific proteins. Here on the left side, this is a section through a hippocampus that's been stained with NuN, which is a neuron-specific marker. That's the brown labels. All of those brown cells are neurons. You can see that there are other cells that have been counterstained with Nissl stain that are not neurons, presumably. They're glial cells. Similarly, we can do this with fluorescent labels. This image here is showing us neurons in green, and the red is showing us a protein called glial fibrillary acidic protein, which is part of the cytoskeleton that you find in astrocytes. And you can see the structure of the astrocyte here surrounded by neurons. In recent years, the advances in bioinformatics and biotechnology have made it possible to do analysis on a large scale. One of the best examples of this is work that's being done at the Allen Institute. They've taken on the goal of trying to identify the genes that are expressed by particular cells throughout the entire brain, both in mice, which is what I'm showing you here, as well as in humans. This work has generated a huge database which you can search in order to find the expression pattern of genes of interest. For this video, I decided to look at some neurotransmitters. So first I searched for a gene that's expressed only in glutaminergic neurons. This is a vesicular glutamate transporter gene. That's what's shown on the top. And then I also searched for the gene encoding GAD, the enzyme which is necessary to synthesize GABA. For each of these, I'm showing you only one section from an entire brain that was stained to show what cells are expressing these different proteins. We can zoom in on the structure and look to see if there are any differences, and there are. Let me orient you. I've taken a blow up here of a region between the thalamus and the cerebrum. So here's the major fiber tract. This is the internal capsule, and over here is the thalamus, the striatum, and the cerebral cortex. Similarly for the GABAergic section, the thalamus, striatum, and the cerebral cortex. While both pictures show us that the brain contains many glutaminergic neurons and many GABAergic neurons, we can start to see some differences. So for example, deep in the thalamus, there are many glutaminergic neurons, but few GABAergic neurons. Not all proteins are expressed at such high levels everywhere. Here's a different neurotransmitter. This is neuropeptide Y, which is a peptide neurotransmitter. And what I want you to notice is that while there are some cells in the cortex that are expressing neuropeptide Y, by far the most intense staining is in a particular spot, this arcuate nucleus of the hypothalamus. Here's another example of a restricted staining pattern. Here I'm staining for dopamine by looking for the expression of the tyrosine hydroxylase gene necessary to produce dopamine. The staining is restricted to this area in the midbrain, the ventral tegmental and substantia nigra areas. And finally, here's an image for serotonin, and I've identified the expression here by tryptophan hydroxylase expression. This is a section that's further back in the brain, and what I hope you can see is that there's a series of nuclei that are staining for serotonin in the midbrain, down in the pons, and in the medulla. And collectively, these are called the Raphe nuclei, and they're the site where serotonin is produced. So more or less all of the cells that are producing serotonin lie in this deep cluster of nuclei in the brainstem, close to the midline. It's also possible to make transgenic reporter animal lines. Now, these are going to be mice, or they could be some other kind of model organism, which is stably expressing a label for a particular region. Here, for example, we have a line on the left, which is expressed only in the dopaminergic neurons of the ventral tegmental area and the substantia nigra. On the right, we have a line that's stably expressing a protein in the Purkinje cell layer of the cerebellum. Let me close by telling you about one more technique. These are Brainbow animals, which have been generated in order to help study connectivity in the brain. And what we're looking at here are images that have been taken through various sections of the brain, the hippocampus, a cross-section through the nerve tract and the brainstem, and these motor neurons that are innervating ear muscles. Each of the neurons is expressing a unique color. And this has made it possible then to look at a very crowded area like the dentate gyrus of the hippocampus and identify individual neurons even if they have overlapping patterns. The staining techniques I've shown you in this video have ranged from relatively simple to really quite complicated. There are many neuroscientists that go through their entire career and never look at tissue stained with anything more complex than a Nissl stain. But for other neuroscientists, those who are interested in studying the brain at the level of individual neurons and their interconnectedness, advances in cell and molecular biology have made possible experiments that Cajal and Nissl could only have dreamed of.